The overall goal of this procedure is to study neuromuscular junction functionality by means of an ex vivo experimental approach. This is accomplished by stimulating the muscle nerve preparation in two ways, directly on the muscle membrane and through the nerve. Since membrane stimulation bypasses neurotransmission signaling, any differences between the two contractile responses can be considered as an indirect measurement of neuromuscular junction functionality. Here we present this procedure in soleus-sciatic nerve preparation. The muscle is dissected together with its nerve and placed into a perfused tissue bath. It is fixed to a force and length controller, and a platinum pair of electrodes are placed parallel to the muscle. A glass suction electrode is then moved close to the cut end of the nerve. A comprehensive testing protocol is then applied to thoroughly evaluate both neuromuscular junction and muscle functionality.
The functional connection between muscle and nerve is crucial for both partners to survive and function. And the first region at the level of which the two tissues communicate is the neuromuscular junction, which normally displays a pressure-like morphology. Nevertheless, in several pathologic conditions, the functional interplay between muscle and nerve is severely compromised, and neuromuscular junction loses the complex morphologic organization. The overall goal of our procedure is to study neuromuscular junction functionality by using an ex vivo experimental approach. This is a